We're going to continue on with a review of enzymes. Enzymes are catalysts that speed up the rate of chemical reactions by decreasing the activation energy. And remember, they don't affect the direction of the reaction. So if it's an uphill endergonic reaction, it's still going to go uphill. Enzymes are usually which type of organic molecule? So remember that most enzymes are going to be made out of proteins. And if you remember, the enzymes have an active site. That's where the substrate will fit into. And then the enzyme will catalyze the chemical reaction, form the product. And then the product will come out of the active site, and then the enzyme will be ready to use again. And we'll accept another substrate and then do the whole reaction again. So this graph just shows the activation energy with and without an enzyme. So the line in red shows the activation energy without the enzyme, and then in gray is the activation energy with the enzyme. So you can see that the enzyme will lower the activation energy, and that's why it speeds up the chemical reaction. But you can see that the change in free energy, the delta G, will remain the same. So the reaction will still go the same direction. Like if this is a downhill exergonic reaction, it will stay that way. So enzymes are very specific for which substrate they're going to bind to. And that's because they're very specific in their shape of their active site. So remember that they're made out of proteins, and they have the up to four levels of folding, and it's like a lock and key for the substrate to fit into the active site. And you can recognize an enzyme because most of their names will end in ACE. This picture just shows how specific the enzyme active site shape is. And you can see that substrate C is the only one that has the right shape to fit into this enzyme's active site. So the other substrates here will not fit into the active site for this particular enzyme. A lot of enzymes will require a cofactor or coenzyme as well to work. The difference between these two: cofactors are inorganic metal ions, so they could be, for example, a magnesium or copper, zinc, or iron atom that will attach to the enzyme, usually at a site away from the active site, and it will change the shape of the active site. So now it's the right shape to be able to bind to the substrate. Coenzymes are organic, and a lot of times they will be B vitamins. And they serve the function of transferring chemical groups or electrons during the chemical reaction. So some examples of coenzymes would be coenzyme A, FAD, NAD+. Those are all coenzymes. Here's a picture of a cofactor. So maybe this is a magnesium atom that is attaching to the enzyme at some spot away from the active site, has changed the shape of the active site, and now that allows the substrate to bind, and now the enzyme will work. And here's a picture of the coenzymes. So the coenzyme oftentimes will fit into the active site, and it's blue in this picture. And it will transfer chemical groups or electrons during the chemical reaction. So there's a couple of factors that will affect the rate of enzyme-catalyzed reactions. So there's just how fast the enzyme catalyzes their reaction. So some enzymes just work faster than others. So that's just a property of the enzyme itself. There's the substrate and enzyme concentration. That's something that the cell can change. The affinity of the enzyme for the substrate, that's just, again, a property of the enzyme. And then temperature and pH, usually the cell is not going to be changing those because we want to maintain those in the homeostasis, so we don't want to usually be changing those. Let's take a look at substrate concentration. So if we take a look at this picture, you can see that as you increase the amount of substrate and add more and more substrate, more and more of the substrate will bind to more and more of the enzyme until you get to a point here where all of the enzyme are bound by the substrate, and even if you add more and more substrate, there's no more enzyme available for the substrate to bind to. So we say the enzyme is saturated. And so even though you're adding more and more substrate, you can't go any faster with the reaction rate. You can't make the product any faster because the enzyme is saturated. So in order to get out of that saturation and to get the reaction going faster again, the way that you would have to overcome that is if you added more enzyme. And then you could bind to more of that substrate, and then you could get the line going up again and go faster again. This is showing the concentration of the enzyme. So if the cell makes more and more enzyme by turning on the gene to make the enzyme, you keep making more and more of it, then you can bind to more of the substrate and produce more of the product faster. So as you increase the concentration of the enzyme, the reaction rate will increase. So that'll be our answer here. Okay, each enzyme has an affinity, or how well it binds to the substrate at its active site. So for example, we've got this particular enzyme on the left with its active site, and we've got two different substrates here. And you can notice that the one on the top, the green substrate, actually will bind better to the active site based on the charges than for the purple substrate. So the enzyme will have a higher affinity, will bind better to the green substrate than it will to the purple one. And so if we look at a graph, you can see that you're going to get a faster reaction rate with the green substrate than you would with the purple one. Here you can see the effect of temperature on an enzyme's reaction rate. So our enzymes are designed to work best at our body temperature at 37 degrees Celsius. So you can see they work the fastest there and produce the most product. But if our body temperature starts getting too hot, you can see that the enzymes start to slow down and don't work as fast. And what's happening as your body temperature starts to get too hot is that the enzymes being made out of protein, they start to denature and unfold. And when they denature, the active site shape disappears because it's unfolding and it can no longer bind to the substrate, and then you can't do that reaction anymore. And then on the opposite end, as you cool down too much and get too cold, the reaction rate from the enzyme also slows down here too. And what's happening there is that the molecules start to move slower in the cells. And so everything is just moving in slow motion. and the enzymes are moving in slow motion, and so the reaction rate really slows down. And then on the right is the effect of pH on enzyme speed. So you can see the enzyme pepsin. That is the digestive enzyme secreted by the stomach, and that starts protein digestion in the stomach. And you can see that it works best at a pH of 2, which is very acidic. And if you get more acidic than a pH of 2 or more basic or less acidic than a pH of 2, it really doesn't work as well. So it really starts to slow down. So what happens when the food moves from the stomach into the small intestine, the pH of the small intestine is much more basic or alkaline. So when that food moves into the small intestine, where the pH is around 8, then you've got to secrete another digestive enzyme to continue the protein digestion, because what happens when you get out of the range of pH for the pepsin is it denatures if you're not at that optimum pH, and that's why it slows down. So in the small intestine, the pancreas will secrete trypsin, which you can see works best at a more basic pH, and then that will finish digesting the proteins that you've eaten. So if we answer our question here on the bottom, what happens to the enzyme shape as you move away from the optimal temperature and pH? It denatures. Okay, and we'll say if too hot. It doesn't do that. It doesn't denature if it gets, if your body starts to cool down and get too cold. Just things are moving in slow motion. So it does denature though if you're getting heat stroke though. Okay, and then when we do metabolic reactions in our cells, there are pathways, and they can get pretty complicated. There's usually multiple steps till we get to our final end product, and a lot of times the metabolic pathways will even branch and go down separate pathways, and you can end up with multiple final products at the end. And each of these steps in these metabolic pathways is going to be catalyzed by a different enzyme. We do need to regulate enzyme activity because conditions will change in the body depending on what you're doing. So if you're sleeping versus exercising, you're going to need to be turning on and off different chemical reactions in the cell. So how we do this is by regulating the enzyme activity, and that's going to control, what chemical reactions are being turned on and turned off at a particular time. So there's a couple ways that we can regulate enzyme activity. One is you can change the enzyme concentration by turning genes on or off, but that takes some time. So quick ways to turn enzymes on and off would be allosteric regulation, covalent regulation, feedback inhibition, and feedforward activation. So let's take a look at allosteric regulation first. So what happens in this is if we look at this picture, we've got our enzyme here with its active site, and somewhere on the enzyme away from the active site, we've got this allosteric site. And that's where the allosteric inhibitor or allosteric activator is going to bind to the enzyme. And when the allosteric activator or inhibitor binds to this allosteric site reversibly, it will change the shape of the active site. And if this green thing is an allosteric inhibitor, it will change the active site so then the substrate can't bind. And that'll turn the enzyme off. And then if this green thing happens to be an allosteric activator, it'll change the shape of the active site so now it will bind to the substrate and that'll turn the enzyme on. And these allosteric modulators bind reversibly. So they'll bind, fall off, they can rebind. So they're just there reversibly. So it's a reversible on-off switch on these enzymes. And then this is just showing you a graph, just showing you that if you have an allosteric activator bind to the enzyme and change the active site shape so now it can bind to the substrate, that'll speed up the reaction and you make more end product. But if you have an allosteric inhibitor bind to the enzyme, that'll change the shape of the active site. So now the active site is the wrong shape, it doesn't bind to the substrate, and that will slow the reaction down. Covalent regulation, this is new. I don't think you've learned about this before, but this is just another on-off switch for the enzymes. And this is, we're going to be taking a chemical group, a lot of time a phosphate group, and covalently binding it with a covalent bond to the enzyme, and that'll change the shape of the active site and either turn the enzyme on or off. All right, so let's take a look at this one. So we've got our enzyme here and we've got our active site, and we're going to take our chemical group, let's say a phosphate group, and let's say that in this shape the enzyme is turned off. And we're going to use another enzyme to put the phosphate group on to this first enzyme. So we're going to attach the phosphate group with a covalent bond to a site other than the active site, and notice how that changes the shape of the active site. And now it can bind to the substrate and we've turned the enzyme on and we can do the chemical reaction. And now let's say that, okay, conditions have changed, now we want to turn this enzyme off. So now we're going to take another enzyme and we're going to dephosphorylate or remove that phosphate group. So this enzyme will chop that phosphate group off, that enzyme, and then that will change the shape of the active site back, and now it can no longer bind to the substrate, and that'll turn the enzyme off and stop the reaction. Okay, so the enzymes that will phosphorylate and dephosphorylate these enzymes. So these are really important to know. These will likely show up on the exam. So a protein kinase enzyme. This is going to phosphorylate the enzyme. So it's going to add the phosphate group on. So here we've got our enzyme and we've got a phosphate group. The protein kinase enzyme will grab both of these and stick the phosphate group onto the enzyme with the covalent bond and change the shape of that active site, and maybe that will turn the enzyme on. Okay, and then we have another enzyme called phosphatase, which you should know. And the phosphatase enzyme is going to do the opposite, and it's going to dephosphorylate. So it's going to remove the phosphate group. So it will take that enzyme that has the phosphate group on it, chop the phosphate group off, and now that enzyme is dephosphorylated. That changes the shape of the active site, and then maybe that can no longer bind to the substrate, and maybe that turns the enzyme off. Okay, so here's a picture of this. We've got on top, we've got our enzyme turned off, And let's say that we want to turn it on. So we've got our protein kinase enzyme. It actually will grab a phosphate from ATP, chop it off there, stick it onto the enzyme, and that'll change the shape of the active site. So now it can bind to the phosphate, bind to the substrate. And now we've turned the enzyme on, so we can do our chemical reaction. And now later on we decide, okay, we don't want that chemical reaction going on anymore. Now we need to turn this enzyme off. So phosphatase enzyme will come over and dephosphorylate the enzyme. So it will chop that phosphate group off the enzyme. That'll change the shape of the active site, can no longer bind to the substrate, and now we've turned that enzyme off and the chemical reaction can't go anymore. So this is covalent regulation of an enzyme. So phosphorylating and dephosphorylating these enzymes is just an on-off switch. So turning these enzymes on and off. Okay, feedback inhibition, I know you've learned about this before. So this is just where you've got an intermediate product in the metabolic pathway will feed back to an enzyme somewhere in the pathway beforehand. It acts as an allosteric inhibitor. So it binds to the enzyme somewhere other than the active site and changes the shape of the active site, can no longer bind to the substrate, and that will turn the pathway off until this intermediate product C falls off enzyme 2 again, and then we can turn the pathway back on again. End product inhibition is just a form of feedback inhibition, and specifically it's just the end product here, D, feeding back to the first enzyme in the pathway, acting as an allosteric inhibitor, binding to a site other than the active site on that first enzyme, changing the shape of the active site, can no longer bind to that substrate A, and then that will shut down the pathway until D falls off enzyme 1, and then we can turn the pathway back on again. And then feedforward activation is when you have this intermediate B, for example, feeding forward and binding to this last enzyme in the pathway, and it's going to act as an allosteric activator and will change the shape of the active site so it can bind to the substrate, and we'll turn that enzyme on, and then we can do that reaction and turn that pathway on.